Okay, welcome to video number 27 of the Diaries of a Coronavirologist YouTube channel. Today is the 30th of June, second half of the year just about, and we are up to about 10.4 million confirmed cases of COVID-19, and that's been responsible for about 508,000 deaths. So today in this video, I want to talk a little bit about some research that made the news last week, looking at the waning antibody response, or suggesting that there is a waning antibody response in people who were infected with SARS2. This made the news, rightfully so, as it raises some concerns about herd immunity and vaccine strategies and things like this that I want to talk about, and talk about the implications and why maybe it's not the end of the world for things, but why it's somewhat concerning to see, and just to break down some of the papers that were making the headlines. So the first of these papers, and the one I'll spend most of the time on tonight, was published in the journal Nature Medicine last week. So this is a peer-reviewed journal. It's gone through all the peer review process and it's out. As always, I'll put a link down to the papers I discussed tonight in the description for this video for those who want to go to the primary sources. And ultimately what this paper did was it looked at 37 patients who were found to be asymptomatic for COVID-19 but tested positive on the PCR test, and compared them with 37 largely matched symptomatic patients, so sex matched, age matched, comorbidity matched, to try and have a pretty equivalent population, and look to see how the responses of those people, patient populations, were different. So the way that the asymptomatic patients were found was through a large amount of contact tracing. So the big bulk of this study was performed in Guangzhou in China. Apologies if I butcher any of the pronunciation on the Chinese places. And they were looking directly for asymptomatic people. So they had a lot of confirmed cases. I think they looked at around 2,000 cases total, and then looked at the close contacts, so the people that were in contact with all of those cases, and looked to see who's truly asymptomatic. They initially started with 60 people who claimed to have had no symptoms within the past 14 days since the contact tracing effect took place, and they were positive on PCR for the virus but did not show any symptoms. So those 60 people were then isolated in hospitals, so a centralized location that could be controlled, and further assessed. And over that period that they were being assessed, there was a dropout of 17 people because they were found to have mild symptoms when they were clinically assessed in the hospital. And a further 6 people developed symptoms themselves and reported that they were having symptoms while in that hospital, leaving with 37 people that were deemed as asymptomatic. And this was all taking place around February in Guangzhou. So there's 37 people there. There's a median age in that group of 41, and 22 of them were female. And they were put together with a cohort of 37 patients of matched age and sex and comorbidities, as I've already said. So a pretty comparable population to look at the difference between these asymptomatic and symptomatic patients. So the first interesting thing about this asymptomatic cohort is the fact that when they were assessed in the hospital by CT scans to look at their lungs, there was actually some evidence of lung abnormalities. So these ground glass opacities and stripe shadows that show up on the scans, which are indicative of issues in the lungs, were apparent in these people who had no symptoms. So there are clinical signs of disease, even though there's no symptomology, which is something that has been reported elsewhere. So even though people haven't got overt symptoms, they are still potentially developing some kind of lung damage, which is just an interesting feature of the infection with this virus and something that seems to be somewhat common. So I just wanted to point that out at the front. So there were 10 people who had the ground glass opacities, and 11 who had the striped shadows, and 16 who had no abnormalities at all. So it's not that all of the people developed some kind of lung problems, only a subset of these 37 patients, but interesting that there are clinical signs even in the absence of symptoms. So now that the study has these two groups, this asymptomatic and the symptomatic, they set about to compare the antibody responses that were generated in these people. So just as a quick word, because it will come up in the next paper as well, The main antibodies that are looked at for this virus are IgM and IgG. So Ig is immunoglobulin, a different term for antibody. And there's different subsets of antibodies with different letters. So IgM and IgG are just two different subsets of antibodies. Typically, IgM antibodies seem to come up faster within the first week or so of infection, and IgG antibodies come up a bit later, typically take about 2 weeks. So having more IgG is possibly indicative of the fact that someone was infected a longer time ago. But again, they come up much later in infection compared to when you may be positive for the RNA tests. So again, for previous things I talked about on the channel, for those watching regularly, that's the differentiating factor between the serology test, which looks for IgM and IgG antibodies, and the PCR test, which is more just an active infection. So they found pretty comparable numbers in the two groups. So initial assessments, about 81% of patients were positive for IgG. So 30 out of the 37 asymptomatic and 31 out of 37 in the symptomatic group were positive for IgG. An interesting thing there is that not all the patients had developed their IgG antibodies or did develop the IgG antibodies. And it was unclear to me about whether those patients did go on to or not, but that will come up in the second paper where it looks like maybe not everyone develops antibodies, which is interesting and something I will come back to when sort of discussing broader applications or context for these papers. So those tests were, I say the initial tests, they were around 3 to 4 weeks after the supposed exposure to the virus. So remember that the symptomatic people and the asymptomatic people were through contact tracing. So they knew when someone was infected in a household, for example, and so then they could work out it's about 3 to 4 weeks after the exposure to that, and they still hadn't developed symptoms. So not all people seem to be developing antibodies. That's the first thing. What was also interesting on looking at the IgG here is that the level of IgG was significantly higher in the symptomatic patients. So it looks like if you develop symptoms, you might actually develop a larger antibody response, which is somewhat interesting. And in terms of the IgM, it was again fairly similar. So 23 out of the 37 asymptomatic patients developed IgM or had IgM positivity, and 29 of the 37 symptomatic patients were positive for IgM. So that was just the initial part of the assessment, but they then followed up with the patients 8 weeks after this initial testing. So this is sort of going towards 2 to 3 months after they were initially exposed to the virus, to look at how the antibody response is sustained, or as they found out, how it wanes. So somewhat worryingly, the study found that the levels of IgG declined in 28 of 30 asymptomatic and 30 out of 31 symptomatic patients. So obviously those numbers are the lower numbers because not all of the 37 people developed the antibody response. So 20 out of 30 and 30 out of 31 showed a decrease in antibody, and the median average decline was between 70 and 75%. So even in just 8 weeks, it looks like the antibody response is going down quite substantially. And they also saw a decline in the level of neutralizing antibodies. So these are the ones that are of, I guess, most interest, really, in a sense. So neutralizing antibodies are the ones that we want to generate with vaccines because they're the ones that will neutralize an infection. So the way that this is worked out generally is by the way we do it in the lab here, for example, is we'll get antibodies that we're testing, we'll mix them with the virus for an hour, and then see if we can infect cells with that virus, and we try and dilute out how much antibody there is. So obviously the higher the content of neutralizing antibodies, the lower the dilution, or the more dilute our sample can become before the virus can infect cells. And the way those antibodies work is they'll bind to spike and stop spike's ability of binding to the receptor. So they directly neutralize infection. And this study found that there was a decline in the neutralizing antibodies, but not as substantial as the overall decline. So there's an 8 and 11% median decline in levels of neutralizing antibodies in the asymptomatic and symptomatic respectively. So there's an overall general decline, and there is a decline of neutralizing antibodies, but it doesn't seem that there's this massive drop off in neutralizing antibodies in the 8 weeks. But obviously, it would be better if it was sustained for longer, because 8 weeks isn't exactly a long period of time. So that's pretty much what I want to say on that first paper. There's a few other things in there, but I just want to focus on the antibodies largely for what I'm talking about tonight. And so the other paper that I saw also making the headlines along with that first one when I was looking at the news coverage last week was a paper that was put up on MedRxiv. So I've talked multiple times on the channel about bioRxiv. MedRxiv is a similar thing. It's a preprint server. So the papers haven't been peer reviewed. They're just out there faster because peer review takes time. So it's a way to get data shared quicker, but it hasn't been assessed in the same to such a level as papers that go through peer review. So this paper was again looking at antibodies and looking at them in around February and April time. So between the 29th of February and the 29th of April was when this study was being performed to look at antibodies. So they have 4 cohorts in this study, which were patients that were hospitalized for COVID-19, healthcare workers in hospitals dealing with COVID-19 patients, general workers, and other patients in hospital but not for COVID-19. So this was done in multiple sites. There were 3 different hospitals that were used for the patient cohort, and there were 1,470 COVID patients that were analysed. The healthcare workers were all in the same hospital, which was one of the 3 that were used for the patients, and there were 3,832 of those. The general workers were all in Wuhan, the epicenter of this outbreak, and I'll come back to that momentarily. There were 19,555 general workers that were assessed, so a very large cohort there. And there were 16 other patients that were analysed, but not for COVID, so in hospital for other reasons. So there's a lot of people being assessed in this study, which is good because in the first one, 37 is a very small cohort. While interesting, it's better if we've got much higher numbers of patients being assessed, which is what this paper is doing. So again, I'll just break the numbers down into the IgG and IgM for what was found. So for IgG antibodies, 90% of the COVID-19 patients tested positive. So again, a very high number, but not all of the patients seem to develop the IgG antibodies, which is interesting. Only 4% of the healthcare workers had antibodies, and this is something I'm going to get to momentarily. About 4.6% of the general workers had antibodies, so pretty similar to the healthcare providers, and 1% of the other patients that were in hospital had antibodies to COVID-19. So it's the people who had COVID-19 really had the highest antibody levels as kind of would be expected. In terms of IgM antibodies, it was about 31% in patients, 1.5% in healthcare workers, 1.3% in general workers, and 0.2% in other patients. So again, similar to what was seen in the first paper, the people who developed COVID-19 have antibodies, but not all people. And this paper made a big deal about the healthcare workers, and this is sort of what got picked up in the media coverage of it. And I'm not sure if it really merited the coverage it received. There's quite a long argument made in the paper that this 4% who have IgG antibodies of the healthcare workers suggests that people don't develop antibodies very well to COVID-19. And they make an argument that because in the end of February and into April, or prior to that, sorry, when the samples were taken was in this February, April, but prior to that, in January and early February, there wasn't much thought that SARS2 was transmitted person to person. At least that was the story originally. And they make an argument that those healthcare workers therefore wouldn't have been wearing PPE and would have been exposed to the virus. So the fact that 4% is it's only 4% who have antibodies suggests that you don't develop good antibody responses that are long-lived. Now, my problem with that is there's a huge assumption being made that they would have all been infected or just because they were being exposed. I'd like to assume that if you're dealing with a patient in a hospital who has any kind of respiratory infection, regardless of what you know, or symptoms, regardless of whether you know it's a coronavirus or anything else, you're going to be putting on some form of PPE. So they seem to really want to push this idea that the fact that 4% of the healthcare workers have antibodies and only 4% means that they don't have long-lived antibodies. But there's a huge gap there because they have no proof that those people were originally infected. So I find that odd. I'm curious to see what will happen about this paper getting into a peer-reviewed journal and whether maybe I'm just too critical and not understanding why they can make this assertion. Maybe PPE usage really bad in Chinese hospitals, but I don't think that's the case. So I don't think this one can be used as much as the first one. I think the first paper is a really good one because it directly compares symptomatic and asymptomatic patients and looks to see their antibody responses. The second one, I think, just makes a bit of a leap, but I wanted to talk about it in the same video because they were being talked about in the same news articles I was reading last week. With those criticisms said, though, I will add that it's interesting that this was done in Wuhan, and the fact that the general population, the general workers, the 20,000-ish they looked at there, and the healthcare workers had pretty much the same proportion of antibodies is somewhat interesting. It's about the 4% and 1 and a bit percent for IgG and IgM, respectively. So even with PPE and things like that, you would suspect that healthcare workers would be more likely to be exposed. So the fact that the prevalence of antibodies is about the same in the general population is kind of interesting and a bit weird. And maybe that does point towards the fact that the antibody response doesn't last more than a couple of months. But as I say, the criticism I have is that there's no absolute knowledge that those people were ever infected, whether they treated patients either. So those 3,800 people that were looked at in one of the hospitals, I didn't see in the paper how many patients they might have treated. That hospital might have had very few cases. So while it's weird that the numbers are pretty much the same for general workers and healthcare workers, I just think it's a bit of a logical jump to make the argument that this is evidence that the antibody response isn't long-lived, as this paper was being talked about in media. And as they themselves try to make the argument for, I'd want to see much better evidence and think that the first paper did a better job of that. But ultimately, what the papers are suggesting is that maybe antibody responses aren't very long-lived to SARS2. Now, previous work on MERS and SARS1 has suggested that the antibody responses generally last for about a year, maybe 2, and you can still detect IgG antibodies and neutralizing antibodies. So the fact that, at least in that first study, the neutralizing antibodies seem to wane over that 8-week period is somewhat concerning. Now, again, as I've said, that was a small cohort of patients, and it'd be great if we had the number of patients that were in the second study with the study design of the first one, and hopefully those kind of things are being done. I'm sure they are. It's just a matter of time. Obviously, if we're trying to work out if antibodies last for more than 3 months, we need more than 3 months, and this virus has been around for 6 months. So it's a case of taking the samples and actually finding it out. So I don't want to jump yet to say that the antibodies wane to this virus quicker than any other coronavirus. I don't think the data is strong enough yet, although it is suggesting that maybe it does wane a little bit, which is a concern, at least for antibodies. The important thing, though, here is that antibodies aren't the only part of the immune response to this virus, and importantly, the adaptive immune response. So just because I want this channel to always be accessible to everyone, the immune system has 2 different types, 2 different arms that are linked. So there's the innate immune response and the adaptive immune response. The innate immune response is a very rapid response. It's when you first get infected, you get this massive release of inflammatory proteins that try and suppress viral replication as quickly as possible. So it's the kind of things that give you a fever is the immune response the innate immune response triggering. As those inflammatory proteins are released as part of the immune system, that then triggers the adaptive immune response to kick into action, which takes a bit more time. But that's where you develop this immunological memory and can therefore be immune to reinfection by the same virus. So in the adaptive immune response, it's generally broken down into the B cells and the T cells. B cells are the ones that produce antibodies. So the first thing to say, the antibodies seem to be waning. There seems to be a decline in the amount of antibodies in the COVID-19 patients that are detected. But that's not to say necessarily that the B cells themselves have gone. They might just not be producing antibodies. So there could be a good memory B cell response that could trigger when there's when SARS2 comes to reinfect those people. But the antibodies may not just stay at high levels. Somewhat speculative, but a potential thing to consider. So it may not be all doom and gloom. The other important aspect, and why I'm talking about the two different types of adaptive immune cells, is the fact that these studies are just looking at B cells. And it's possible that the T cells may be even more important for a protective immune response against SARS2 to protect against COVID-19. So this whole part of the immune system that isn't looked at in these two papers. So it could be that there's actually a really good T cell response, and those people, even though their antibodies have gone down, are still protected from reinfection. So these were sort of painted as maybe we can't be immune to this virus, maybe everyone's going to get reinfected in doom and gloom news coverage of it. But there are caveats to that, in that there are parts that are being missed. This is just looking at antibodies. It's not looking at the cellular response about whether there's still memory B cells that could then produce antibodies. And it's not looking at all at the T cells. So not all doom and gloom. And also I want to point out, because I've talked about antibodies quite a lot with regards to vaccines. Vaccines are generally trying to produce an antibody response, but they can also produce a T cell response. And it's also important to point out that vaccines can produce better immune responses potentially than the natural infection. It's the use of adjuvants, which can boost a vaccine's effectiveness. So just because there seems like there might be a waning immune response, an antibody response, I should stress, doesn't mean that vaccination isn't going to work, and it doesn't mean that these people can be reinfected. As I've sort of alluded to, all of this, we need more time. We need to see how long the immunity is. We need good data about whether people are being reinfected or not. And generally, it seems, I've not seen any compelling evidence to suggest that people are getting reinfected, suggesting that maybe they are indeed developing protective immune responses, at least in the short term. And with vaccines, we can potentially develop a better immune response to protect people down the line. So it's not all doom and gloom, even though headlines of waning immunity suggest maybe it is. Anyway, just wanted to cover those stories a little bit, because they were obviously making the news last week. And I just didn't get around to doing a video on them last week, so I thought I'd start the week doing them today. Anyway, as always, if you're finding these videos useful, interesting, please share them with people who might get something from them. Please subscribe and hit the bell so you get notifications when I post, and put a like on the video for the YouTube metrics, all those kind of things. And please leave questions and comments on the videos. I will start trying to get back to looking at them and incorporating them into videos. So if people have questions and things, put them in, and I'll try and tackle them as a follow-up in a future video. And anyway, that all said, as always, please stay safe, wash your hands and wear a mask, and keep calm and carry on. We will get through this pandemic.